Thank you so much for the introduction, Antonius. And yeah, I'm happy to be here. Thank you, everyone, for joining us. I will just talk today about the neuroanatomical changes in gray matter due to tinnitus and hearing loss. And we focused on the military population. A little bit of a background. So, why is it important to examine the gray matter? So, that's because it offers us crucial insights into conditions, underlying mechanisms. And there has been previous studies which were examining it, not on military population to my knowledge, but what they found in civilians was that both the sensory and emotional systems are involved. So tinnitus is more than just auditory processing. And why we focus on the military population, that's because our rationale was that military population really suffers from it. We can see that among the veterans, their hearing loss and tinnitus are the top two service-related disabilities. And we were also thinking that if we are investigating in a homogeneous population, it might require from us a bit smaller size of population. All right. So how did we do that? We collected the data in Texas. We had data from 68 participants. All of them were military population, and their age range was between 21 and 64 years old. And we divided them into two main groups. So we had controls, those who didn't have hearing tinnitus, and tinnitus participants who had that. And later on, we divided them into four subgroups, depending on if there was additional hearing loss or not. And all of them underwent the standard structural MRI. Um, to analyze this data, we used the voxel-based morphometry. And that's a basic tool which examines the gray matter volume. We also measured the hearing of all the groups and subgroups, and that is illustrated on the audiogram. Uh, yeah, and so what we found, um, firstly in the region of interest analysis, so that is, we put a mask on the auditory regions that we were the most interested in. And we compared the group with tinnitus to the group without tinnitus, so our controls, and we found the difference in the thalamus proper. And that has been seen before in Muhlav's paper in 2006. But interestingly, he found the increase in the thalamus proper. And that could be possibly explained by that he didn't account for hearing loss. He was only looking at the tinnitus participants at the moment. We also looked at the whole brain gray matter, so without the prior hypothesis. And that was, then we seen differences in precuneus, superior frontal gyrus, and right planum temporal. And these regions, so precuneus is part of the default mode network, and that one region is crucial for the self-awareness and consciousness. And superior frontal gyrus, on the other hand, is responsible for the executive functions and attention control, and right planum temporal is involved in auditory processing. So we see here that combined effect of tinnitus and hearing loss is impacting both sensory regions like thalamus, but also emotional regions. But when we look alone at tinnitus, that then we've seen difference in the right subcallosal area, and that area is a part of a limbic system. So that's more associated with the emotional regulation. Um, And later on, we also looked at the hearing loss alone. And then we saw that we found those three regions which distinguished between people with hearing loss and without hearing loss. And that was right planum polare and right precentral gyrus and left fusiform gyrus. Right planum polare is the region which is responsible for the pitch perception, and the right precentral gyrus is primarily a motor region, and the left fusiform gyrus is responsible for object and facial recognition. But based on that, we can see that since there is a decrease, there is a deprivation, and it's causing this plasticity-driven changes. And yeah, so what does it really mean? So first of all, we hypothesize that the thalamus plays an important role in both hearing loss and tinnitus. And we also hypothesize that based on most our results, which are on uncorrected level, not on a corrected level, that we might require a larger sample, even if our population is homogeneous, to find those effects of tinnitus or hearing loss or their combined effect to be significant. And we actually looked also at effect sizes recently, and estimated the sample sizes that might be required for that. And we found that to see effect sizes between people with tinnitus and without tinnitus, we might require up to even 9,000 people, at least on the population that we had. If we're talking about whole brain. And if we want to see in hearing loss, then we would need around 800 people. So it also tells us that gray matter differences might be easier detected due to hearing loss, which might directly cause this deprivation and decrease of the gray matter volume. And there is also this interesting relationship where we see if someone has both tinnitus and hearing loss, and comparing to people who don't have tinnitus and don't have hearing loss, we even need larger numbers. So it's really interesting to see that it's not necessarily the fact that if you have both, your brain is going to be gray matter volume is going to be decreased more, but there is something more happening there. Possibly tinnitus is sort of compensating for hair loss, but we still have to analyze to understand that better. And as far as it goes for the region of interest, for that, since we have the prior hypothesis, we might need smaller sample sizes. So we estimated that to see the differences between tinnitus and controls, we would need around 5,000 people. And to differentiate between hearing loss and not hearing loss, only 50 people. And then if it's both tinnitus, hearing loss, and controls, no hearing loss, around 200 people. So that again tells us that hearing loss might be the main effect driving this structural changes in our brain. And also, if we have a prior hypothesis, we have stronger statistical power. So it's definitely recommended to do to have prior hypothesis and use a mask to see those differences. We also want to raise an interesting point, which is the age. So the prevalence of tinnitus and hearing loss is correlated with age. So it raises an important question. So when we try to control for age in gray matter studies or even white matter, we might miss some structural changes due to tinnitus. So we included both analysis with and without age to see how that impacts, and we are mostly seeing effects when we don't correct for the age. And when we don't correct for the age, we see the combined effects of tinnitus and hearing loss in the structures like precuneus, which has also been before reported by Dr. Moring. And yeah, the that's basically what I had. So I guess the takeaway from this is it's good to have a prior hypothesis because it can it's more likely that we can find some differences, and that so far as we see gray matter differences are easier to be found due to hearing loss than to tinnitus. At least there is this more straightforward relationship that hearing loss will cause the decrease. Whereas for tinnitus, some studies report decrease, some report increase. So this relationship is not as straightforward. And yeah, that's I want to acknowledge everyone who collected the data, and we're also very thankful for everyone who participated and for the funding we got. And thank you all to that you attended.